I will briefly present you the objectives and approach that we follow through the project for the development of the roadmap. The theme concept paper, I won't go in detail on it because it's exactly what Claudia said. So it was really our lighthouse along the entire project. So because it was setting our final objective that we wanted to achieve with the roadmap. And we had to keep it in mind from the very beginning to the very end to remember what were EFSA's priority for the next years. In concretely, what are the objectives of the roadmap? First is to define priorities for the integration of omics and associated bioinformatics approaches in food safety. How to do that in practice? We identified some working areas that require further investment to ensure that EFSA's vision is achieved. These working areas will be completely developed in the form of future project proposals for EFSA that take into account the opportunity to cooperate with external stakeholders. This is something that we had to take care from the very beginning because we realized pretty soon that EFSA cannot work alone, especially to solve some general challenges that are present in the application of omics in risk assessment. And the roadmap will serve as an evidence-based decision-making tool to plan EFSA's future activities and investments on omics. So it's our proposal. EFSA then will have time to really evaluate and decide concretely based also on other limitation or objectives that it has, what to do exactly with these proposals. We started the project in March 2023. We ended it now in July 2024. The contractor is composed by a consortium of different organizations. Sequenza Biotech is the leading partner with the expertise in bioinformatics. And then there were a number of subcontracting organizations with different expertise. So we had Joseph Casacuberta of the Center for Research in Agricultural Genomics with the expertise, especially in risk assessment. We had Anion Strategic Pharma Intelligence with expertise in toxicology. NRB with expertise more on project management, and Crowe who helped in terms of communication activities. So the entire consortium really contributed to the success of this project. The project was composed of three phases. The inception phase was about the development of the roadmap plan, so how we were gonna work in the next month. So we defined the methodology. We better defined the scope of the roadmap because it's pretty wide, and we really had to clarify what was in and what was out. Then it was followed by the execution phase, which was the heaviest one because it was about collecting the information that was necessary to build afterwards and design the proposed recommendations. And then there was the roadmap definition phase, the last one, where we worked on the list of project proposals to be suggested to EFSA, and to the communication engagement plan, because as I said at the beginning, the inclusion of external stakeholders in the work is something really important for this roadmap especially. The methodological approach, really high level, we started with different data collection activities to research different areas that really helped us to understand what was the state of the art of the application of omics and the application, but also the trends and the future perspective of the application of omics in risk assessment. We had to use different data collection methods because only one was not enough actually to understand. So we used desk research, literature review, but also we consulted stakeholders through a survey, through some interviews, and through a workshop. So this was a really complete data collection approach that allowed us to actually understand how things are going on and what can be the future perspectives. Afterwards, we put together all the data. We analyzed it and we drew further elaboration from the team. We ended up with some project proposals, which are our recommendation on how to reach EFSA's strategic vision on omics. And this is really the core part of the roadmap. The outline of the roadmap. So just for you to know how the roadmap will look like, it's based on, it's composed of 5 chapters. A small introduction that explain you the context and objectives that Claudia has showed us now, and the definition and assumption. So especially to understand what's the scope of the project. Then we covered a bit the methodological approach. Chapter 3 is about the results and discussion. So all these data collection activities and which are the main findings that were really the basis for the development of the recommendations. Chapter 4 with the recommendations, so with the list of project proposals and how they can be categorized and prioritized. And finally, a closing chapter with the engagement and communication opportunities with external stakeholders. Thank you for your attention. I leave the floor to Ricardo, who will go on with the state of the art.
Okay, so good morning, everybody, and it's a pleasure to be here today. And also would like to thank Claudia, Costas, and the entire EFSA group for hosting us today. And also on behalf of Sequentia and the entire group, thank you for the support that you gave us throughout all this roadmap. So it was very nice collaboration. So as Costanza mentioned, I would like to now go a little bit through the state of the art, so the evidence and the main results before we dive into the actual proposals that we will be presented in the roadmap and that Santiago will present to you. First of all, maybe it's important to define some definitions so that we have a common ground, basically. Here you can see the 3 pillars that are at the basis of the roadmap. So we have the research areas, the risk assessment and development areas, and the omics, of course. So when we talk about the research areas, basically we mean all the areas in which EFSA is currently providing its activity for risk assessment. Of course, all of you at EFSA, you're very familiar with this, but to briefly summarize, we have here 7 main areas regarding the plant health and protection management, the genetically modified organisms, the food improvement agents, including feed additives and food contact materials, food and feed contaminants, foodborne zoonotic diseases, animal health and welfare, and finally nutrition, novel food, and nanotechnology. Then building on top of the team concept paper on omics sciences, we have also a list of what were defined as risk assessment development areas. So what are these areas? These areas are defined as broad strategic areas in which developments could really help for the integration of omics sciences into risk assessment. So they were already identified as important areas in which could be beneficial to perform some development. As I said, we built on top of the team concept paper, so you will see that some of these were already presented in the concept paper, like the improving and facilitating the use of genomics and whole genome sequence analysis, the support to the creation, curation, and usage of publicly available omics data repositories as pillars to support a mechanistic-based risk assessment, the incorporation of metagenomics in risk assessment, the incorporation of other omics technologies like epigenomics, transcriptomics, and metabolomics and proteomics. Then in the team concept paper, there was a reference to exposomics, and then we built on top of that to expand it a little bit so that we could, in general, talk about multi-omics approaches for a more realistic food exposure assessment. What is relevant to say here is that these areas could be actually cross research areas. What I mean is that they are not specifically tailored to a specific risk assessment area, but they could actually be applied to different areas. So as part of our activity, we really try to identify the possible interactions between the research areas and what we call the RADAs, the risk assessment development areas, in order to evaluate the possible beneficial effects of these RADAs on the different research areas. And of course, at the very basis of this roadmap, there are the omics sciences. I don't think we need to define them here, but it is important to note that after our activity and research activity, we decided to focus on 5 of the most important omics, which are metagenomics, transcriptomics, proteomics, metabolomics, and genomics. However, I want to point out that even if these are like the major omics sciences in which we see already a great potential for application, or they are already applied in risk assessment, we have not excluded other omics sciences when they were relevant for risk assessment. So we try to be really comprehensive in this sense. So, as Costanza said, we used different approaches and different methodologies in order to, first of all, collect information and map all what is relevant for the application of omics into risk assessment. And as you can see, there was first a mapping activity that, as was mentioned before, included not only literature and desk research, but also an active interaction with multiple agencies. Of course, first of all, EFSA, but also other European risk assessment institutions, also national and including European and extra-European regulatory bodies and international organizations, as well as academia and private sector. And as we said, we used methods like surveys, interviews, and also workshops in order to really get feedback from all these stakeholders. And thanks to this mapping activity, we were able to basically understand what is the state of the art of the omics into risk assessment, and also, very importantly, to identify knowledge and data gaps that could hamper the application on the realization of the objectives of the RADAs that were previously described. And of course, at the same time, also identifying the added value of the different omics sciences into the different areas. Okay. So here, in a nutshell, you can see the very main results that we found and the main conclusions. Of course, here it's just the tip of the iceberg. You will find much more information in the final document. And here we focused on 4 main omics. So you will see from so starting from genomics, you will also see proteomics, metabolomics, and transcriptomics. And then metagenomics and multiomics. And we arranged them in order in following our characterization in terms of the level of adoption in regulatory science. So from genomics, that is already quite widespread, I would say, also already in EFSA, especially for foodborne outbreak investigation and for regulated products, up to the level 4, which are more, let's say, newer or younger omics sciences in which there are not so many examples of applications into risk assessment. So starting from genomics, as I said, we already found that is currently implemented in many, in different areas of risk assessment in EFSA. However, we also found that there is an uneven adoption by member states, and specifically a slower adoption of long-read technologies. In terms of proteomics, metabolomics, and transcriptomics, we see that is being used more and more in EFSA risk assessment, even if it's not yet a routine. However, there is an international and widespread recognition of the potential application of these omics into especially chemical risk assessment, as it is supported by EFSA, other European projects, and OECD, as was mentioned before. Regarding metagenomics, again, there is a global recognition that it could be extremely useful for aiding into risk assessment. However, at the current stage, let's say there are no legal requirements to consider metagenomics of microbiome data into risk assessment. And we see like as a main, let's say, possible limitation, the lack of established guidance for evaluating microbiome-related health effects. Finally, as regards the multi-omics, again, there is a really promising application for these different omics and their integration to have a really complete overview of the biological systems and then to perform deeper risk assessment. However, we have not found many applications of this into actually regulatory science. However, there is a increasing growth usage in research and academic. So there is a potential for application, of course, into risk assessment. As was mentioned before, as part of our mapping activity, we were also able to identify challengers and blockers. So basically, again, blockers that could limit the application of omics sciences into EFSA risk assessment. And we took into account all of them in order when we designed the project proposals that you will hear about. Here you can see the most important ones, like the lack of standardization, validation, and harmonization. Of course, these are broad topics, I would say. They are not necessarily applied to all the omics at the same level, and all of this will be more detailed in the final document, in the roadmap. There is also the problem of poor data integration, the lack of advanced skills among relevant stakeholders. There are limitations sometimes in data access and sharing, as well as regulatory constraints. Regarding the dependence on applicants' information, what happens is that also due to the lack sometimes of guidance and standardization, sometimes applicants are not very uniform, let's say, how they present their results and how they share their information, and this could also limit the broad application of omics. There is a lack of references and updated databases, as well as case studies, and finally the lack of openly accessible detailed methods that could be widespread into the community. Okay, so finally, building on top of all these mapping activity and the challenges and blockers, as I was saying before, we developed a series of recommendations that now Santiago is going to present to you.
Thank you, Ricardo. And first of all, I want to really thank here in this recommendation part of the work that has been done by the EFSA staff, because for us, the risk assessment was a new concept, or basically new for many of us, and it was a really, really collaborative work. So thank you so much for that. And also, I want to thank all the participants in the workshop, because with the expertise, and we try to have a really broad expertise, they help also to define the different part of the project proposal, and it was a really, really helpful for us to dive into this final 6 proposal that we have. So basically, as what Ricardo was saying, this was a really step-by-step activity. So we map all the omics interactions, intersections with the different areas, and then we start thinking about the knowledge gap, what were the challenges, what are the blockers, and building on all of this knowledge through the time. We have identified 6 proposals that, in our opinion, will help EFSA in the next few years to achieve this goal of adopting omics and bioinformatics approach in risk assessments. So here the presentation that I will make is just an overview, in the sense that we try to define the main points of the proposals, but you will find everything in the roadmap, in the final roadmap. And there it is structured in the following manner. So we first have a definition of which is the problem to be addressed. So what we need to solve and for this proposal, what is the need. Then the description of the scope, the objectives, which actions we recommend toward the implementation. Then we evaluate the feasibility and the readiness of the proposal, the expected impact, the collaboration opportunities. As we highlight, as Constanza also has highlighted, it is really important for us and for EFSA. We believe that this is a common challenge between many agencies, so it's an important aspect to highlight. The time frame and the budget, and then a SWOT analysis, identification of the strengths, the weakness, the opportunities, and the threats in each of the proposals. So basically, in the final document, you will have all these chapters well explained for each of them. So basically, in order to try to identify the readiness level and the prioritization of each proposal, we define a few categories. And for the readiness level, which means to try to define which is the readiness, the regulatory readiness of a proposal. So it is ready to really be implemented or not from a regulatory point of view. We have defined a few categories from low to high, meaning low, that is still need a lot of work to be implemented, and high, it is really ready to go on and to be implemented. So then each of the proposal was basically evaluated by different aspects, like the horizontality, and this means how applicable it is through EFSA in different research areas, the research area that was mentioned by Ricardo, the feasibility and the resource. So it is a proposal that needs a lot of resource or not, the urgency and impact, the sustainability and the scalability, risks and uncertainties. So it is good to implement it, but what are the risks of doing that? And then the novelty. So basically, this was the criteria that we use to prioritize the projects. And just to put a high-level overview, we have defined these 6 proposals. So the first one is harmonization of the sequencing and bioinformatics approach. And this involves basically most of the research areas that could have applications in here. But as we said, we try to do this crossing between research areas and risk assessment development areas. And in this case, we really focus in the risk assessment development areas, which the aim of continue improving genomics through risk assessment in EFSA. And we believe that there is a high readiness level. All is ready to go on with this proposal. The second proposal, it is utilizing public Omnic repositories and machine learning models to infer chemical groupings. And here we have 2 research areas. First, it is the use in plant health and also in food contaminants. And here we have 2 risk assessment development areas to be care of. That it is the development and the continued supporting or creation of databases and also the use of other omics. And this is moderate in the sense that there are several challenges that needs to be solved. Then we have the metagenomic proposals that it will promote adoption in EFSA risk assessment. And here, and we will see it later, we have various challenges. And Ricardo was saying, and there is a high interest in, but still there are not many really high-level applications yet in regulatory science. But we believe that there is a work to be done there. Then we have the omic-based risk assessment of GM plants. And here we believe that the application of omic will help, but still need work to be done. And through this proposal, we believe that could be achieved, or at least be more near to the achievement. Then we go to the allergenicity assessment of novel food, and here it is moderate high. We believe that there has been in the last year a lot of work in this direction, a recognized potential of proteomics, that is the omic that we will suggest, and we believe this moderate high. And the last proposal, it is the omics data analysis platform development. And here we believe that EFSA is doing a lot of work in this direction. There is high interest inside the agency to work in this direction. It's aligned with EFSA vision, and we believe that that also is a high readiness level. So now I will go through the proposals. And the proposal number 1, the structure that we present here to try to be like assertive as possible in a few minutes. We have like three big aspects: the current use, the challenge, and the solution we propose. And then we work with the objective, the general objective, action towards implementation, and the collaboration. But again, all of this is found in the final roadmap, and it will be much explained in details. So what we found here is that there is a really well adoption of short-read sequencing And there is a lot of work inside EFSA and other agencies. But we believe that nowadays, EFSA is receiving assemblies that are made with long-read technologies. But there are currently no established guidelines for working with long-read sequencing or hybrid approach. So each of the applicants could have different approach, and this could be a problem that needs to be solved. And also because poor quality assemblies hinder the identification of, for example, novel resistant genes and impede the understanding of antimicrobial resistant mechanism. So basically, there is a need to work through better assemblies, and we believe that there is need for more guidance to the applicants to work on this direction. And as a solution, what we propose is working on harmonized guidance for hybrid sequence approach. And we believe that the hybrid sequence approach is a nice example because it's overcome the specific limitation that has both the short read technologies and the long read technologies. And it is widely recognized that the combination of the both are producing the best assembly. So case-by-case applications need to be evaluated if there is a need of this type of assembly. And also it will be, in our opinion, really good if there is a guidance for doing so. So basically, this is the idea. So we focus first in the harmonization of hybrid sequencing approach for regulated products, because here is where EFSA is receiving the different applications. And through going through this, we believe that there is a need of building case studies. And this was a challenge that was identifying the need of case studies. So we try to implement actions that solve the different challenge. So then the standardization and harmonization of the protocols and guidance, then promoting the adoption, and then providing training. That could be for internal or external stakeholders, is something that needs to be also evaluated. And I will go quickly through the collaborations, but we believe that in many of these areas that are really associated with big challenge in risk assessment, EFSA has the opportunity to collaborate with international organizations, risk assessment agency, European sister agencies, but also with third country risk assessment organizations. And we believe that in case that it is needed, and some of the member states are already doing, if the long-read sequencing is being utilized, will be good also to collaborate with the European reference laboratories and the national reference laboratories. Then the second proposal, it is planned to work in the utilizing the public omic repositories and machine learning models to infer chemical groupings. And what we have seen here is that the omics has potential in predicting chemical groupings, and this is already being discussed inside EFSA with specific tenders. But we found that there is dependency on external maintained database that require continuous updates. So because omic data from public repositories is underutilized in regulatory risk assessment, there is an opportunity here to be fulfilled. And we believe that EFSA is in a position to work in this direction. But there is also a challenge regarding this because there is an inconsistent omic data reporting that complicates the quality evaluation interpretations. So as a solution of these challenges, what we have recommend is to develop and implement machine learning models to infer chemical grouping, enhancing the speed and the accuracy of chemical assessment by leveraging existing omic data more efficiently. So basically, the main idea here is, okay, we have a lot of public omic data, and we need to start using it because there is a really valuable resource, but there is still gaps in the knowledge of how to utilize this type of data, what needs to be done. And that is why we believe that this chemical grouping was a good example that is built on EFSA expertise and what is EFSA right now doing. And with the main goal to have also faster risk assessment. That is also a priority for EFSA. And this is a proposal that is really well aligned to that. So which are the actions that we have planned? First, we need to mine the omics dataset from public source. But it's not just mining, because we need to take care about the standardization of the data, ensure the consistency, the reliability across different datasets. So this is also a main topic. When that is identified, we can develop the machine learning models, test the pipeline, and obviously at the end, train the different stakeholders. And here again, the collaborations are the International European Risk Assessment Organization. This is a topic that all the agency care about, and there is nowadays, and every year is much and much more the public Omics databases that are available. Here we are focusing in chemical risk assessment, but this is true also for the other areas. There is the national, European, and third country organizations, but also here we can get help. EFSA can get help also from the industry, the academia, and also database curators to build more expertise and to be really be integrated with different stakeholders. Then we go through the metagenomic adoption, the metagenomic proposals, and here we design big proposals that tackle different objectives, because what we have found is that there is a general global recognition of the value of metagenomic through different regulatory agencies. Not just EFSA, but different European sister agency, international one, international third country organization. So there is a topic that is really discussed and could be useful for tracking microbial change and identifying new risk independently of traditional culture method. Again, other things. But there are several challenges we work on, and we believe that the EFSA is in a position to start working on that, and it could also guide the implementations. And one of the big challenges is the harmonization required for metagenomic standard from sampling to analysis, so all the steps. And also gaps in understanding the baseline of microbiomes, and also lack of comprehensive database that affect differently in different research areas inside EFSA. So which is the solution that we propose, or at least a solution to help to start working on that direction, is to develop and define comprehensive databases, standards, conduct proof of concept case studies to evaluate and promote the adoption of metagenomics. So our idea here is to have this general proposal and to try to fill some of that gaps, or start working in filling some of that gaps. And for that, what we have proposed is 4 case studies. Again, we are working here in the challenge of standardization, harmonization, in the challenge of case study, in the challenge of comprehensive database. So this proposal is really covering a lot of the challenge, because metagenomics has, until now, has several challenges to be fulfilled. And this was also clear with the work of the workshop. So it was a proposal that tried to cover different aspects that could be helpful for EFSA next generation risk assessment. And we have one of the case studies that is oriented in the detection and characterization of foodborne pathogens. Another one that assesses the technique sensitivity and the specificity of the AMR databases. Other that is the direction of the harmonization of the analysis of the metagenomics analysis. And also one that is tackled to do the profiling of microbial communities in samples with low microbial abundance and high host DNA. So each of these proposals is really, case study is really well explained in the document, but the idea is to try to fill different gaps that exist in the application of metagenomics. And then we go through the actions. And one of the actions that we believe that is needed, because there is we don't have find a lot of information available, but it's reviewing the capacity building and the needs of the member states through metagenomics. There is a lot of work doing for genomics in that area, but we believe that is still need for metagenomics analysis and to adopt metagenomics in the risk assessment. Also, there is we believe that there is a need of a comprehensive review and workshops to refine this case study with different expertise coming from different areas. Then the development of the case studies, and finally, provide integrative guidance and support, because as this is new in the regulatory science, There is not a lot of documentation in the guidance and support, and we believe that through these case studies could be the start of building this type of documents, and then obviously the dissemination of the research. And here, for example, we believe that there is an opportunity to collaborate with standardization organizations like ISO for the standardization of the methods, the OECD, the Joint Research Center, European reference laboratories, European sister agencies, and academia. Then we have designed two proposals to tackle the incorporation of other omics, and here we highlight metabolomics, proteomics, and transcriptomics into risk assessment. And we believe, and in line with what was discussed in the omic concept paper, that the risk assessment of GMOs, and specifically we highlight the GM plants, could be a proposal to do this. And nowadays, omic data are used on a case-by-case basis by applicants to complement a risk assessment of GM plants, but there are challenges on this. First of all, EFSA recognized the necessity for update methods in assessing complex GM plants, and omic technologies, while promising for this, still lack standardization and comprehensive case studies. So this proposal, the idea of this proposal is to tackle that, to standardize omics methodology or to help in the standardization of omic methodology, and to generate case studies to evaluate the use of omic profiles as a robust alternative or complement to current GM plants risk assessment methods. So basically, the objectives is working with the comparative analysis of GM plants, especially those with complex composition, that could be significantly altered due to multiple genetic modifications. And how is planned to do this? First of all, we believe that there is a need to gather insights through workshop or through different activities on the integration of omics in GM plants risk assessment, then work on the development of the case study that could include a well-known GM plant, but also a more complex one. So to have both of the sides. Then we believe that could be an opportunity also to work through the creation of comprehensive database for omics profiles for plant reference varieties. We think this is a second step, and we put it as an optional in the recommendation, but we believe it could be viable. And then obviously work toward the standardization of the omics. And here we think that there is a really clear path of collaboration with the OECD, because as Claudia mentioned at the beginning, they are working on the framework for reporting omics, and especially transcriptomic, metabolomics, and possibly proteomics. So there is a high interest in that framework, and we believe that it is a good opportunity to collaborate and to apply here in this proposal. But also there are other partners that could be achieved, like research institute and other regulatory bodies, to develop these case studies. Then we have the proposal number 5, and this is related with the allergenicity assessment of novel foods. So here again, we are tackling the other omics adoption and risk assessment, and particularly proteomics in this case. And why we believe this proposal is important? Because there is a rapid expansion of novel food that require thorough safety evaluation to protect the European consumers. And nowadays, proteomics is considered by applicants and regulatory agencies and evaluated by recognize the potential for analyzing the novel food proteome. But there is challenge in the novel food safety evaluation, and one of them is that traditional method for assessing allergenicity may not adequately address complexity posed by novel food. And also there is a lack of standardized procedures for proteomics. So the idea is to employ and target proteomics to analyze the protein content of novel food for allergenicity risk assessment. And this could include novel and probably unknown ones allergens. So it's not just the known ones, but also through the proposal, we believe that it is possible to identify novel ones. And doing that, we believe that there is a chance, or there is an opportunity to providing a more comprehensive overview of allergenic risk. So basically, the idea is select a target proof case study, do the proteomics analysis, do the allergenicity prediction, the experimental validation, and then work toward the implementation of proteomics And here again, we have a lot of opportunities and collaboration that are well explained in the roadmap. And the last proposal is the Omics data analysis platform development. And here we have a clear path in this direction that EFSA is currently working. And EFSA has created this analytical framework that utilizes Azure Batch to deploy bioinformatics pipelines, but is designing primarily for specialist use. But there are challenges with this and that can arise that this could be related with the limited interaction and visualization capability for broader stakeholder use. So what we believe that would be a good proposal and a solution for this is to provide a dynamic visualization and analysis, a platform to do that, integrated with the existing framework to improve the usability and data-driven decision-making in line with EFSA vision. So basically, we believe that the main idea is to provide a way to have an easy deployment of the bioinformatics portals that are being built in EFSA and feature different aspects like the user management, the data storage, the connectivity, visualization modules, and all of this with the scope for future technological standardization. So helping EFSA and potential external stakeholders to do omics analysis and visualize and interpret the results. So basically, the action is first evaluate the EFSA framework capabilities, so which is the status right now, then develop the different modules, ensure the security and compliance, and provide training. And then, obviously, there will be a need for implement continuous improvement process because there need to be new portals, there need to be new analysis. So it is a continuous work that needs to be done. And here again, we believe that there is a lot of potential collaboration to this proposal.
Thank you, Santiago. So after having checked the recommendations of concrete projects that could help EFSA to reach its vision on omics application and risk assessment, we conclude with a chapter on stakeholder engagement and communication. This chapter is actually more general because for each proposal, as you have seen in Santiago's presentation, we have identified a specific list of stakeholders that could be engaged and involved in the concrete implementation of the projects. This chapter is actually more general. So in general, when we talk about integration of omics in risk assessment, which are the stakeholders that we have to keep in mind? What can be their role? How EFSA should approach these stakeholders? Methodologically speaking, we started by mapping all EFSA stakeholders. So we try to understand what they do or what they think about the integration of omics in risk assessment. And we identified two big groups. So those stakeholders that actually are already working with omics or they know really well what we're talking about, and those stakeholders that are now pretty far from this concept and where there were just some really few examples of stakeholders that were actually aware of the opportunity given by omics approaches in this field. Afterwards, based on this mapping, we assess them. So to understand what type of relationship should EFSA establish with them when talking about omics application and risk assessment. And afterwards, we designed an engagement and communication plan. So pretty generic, again, but explaining exactly why to engage them, who to engage, why to engage, and how to engage with the relevant stakeholders. We concluded with 3 final recommendations that could be applied overall. So here you can see the list of all stakeholders that we mapped and that we took into account. This assessment provides an indication on how to relate with the stakeholders towards the adoption of omics approaches in risk assessment. It's not a ranking. So it's not what you see here is not that a stakeholder is more important than another. It just tells us, depending on the quadrant they fall into, what EFSA should do in their relation. Also knowing that actually this assessment can change over time. So we see, for example, that there are some organizations that are whose sensitivity and whose perception about omics is changing and is likely to change in the near future. So monitoring them, especially those that in the quadrant on the lower part, basically their role could change over time. And this is why it's really important to monitor and always to inform them about EFSA's and other stakeholders' activities in omics. The others, those that you can see in the right part of the graph, are those that are already pretty relevant in terms of engagement. These are the type of organization that we have already mentioned in our proposals, and some of them are already pretty working a lot with EFSA. So the idea is to maintain and reinforce the engagement and relations in concrete projects, and others that actually could be included more because they are really relevant and could be really impactful. So another concept, what we understand here is that each stakeholder should be engaged in the right way, provided, and we provided additional information on why and how to set up collaboration depending on the quadrant they fall into. The very final recommendation for the stakeholders engagement, in the chapter you will see also some tables explaining stakeholder per stakeholder, why they are important, how to engage them in terms of specific ideas, so in terms of workshop or doing bilateral meetings, forums, so different ideas on how actually to do this engagement. The final recommendations for the communication and engagement of stakeholders are this one. So first, we need to work on general awareness on communication. So developing a comprehensive omics training program accessible via on-demand webinars for EFSA's technical staff and key risk assessment stakeholders, aiming to increase awareness, foster innovation, and serve as a foundation for future omics application. So we understood that actually there is sometimes a lack of awareness of the opportunities that omics can give to risk assessment. So maybe, especially the technical staff, being more aware, getting more awareness on this will also help foster a bottom-up approach. So instead of always using a top-down guide on what to do, also bottom up. So every person working in risk assessment being more aware of the opportunities of omics will help the entire organization to develop a wider omics application in the process. This is really general and also directed not only to EFSA staff, but also to external stakeholders. Afterwards, and this is something that we mentioned in the proposals, there should be the development of advanced application-specific trainings, really connected to the case studies, to the specific projects that EFSA will implement, directed to those that are interested in it or that are actually working on it. Just to be sure that the awareness, but especially the skills, are good for purpose. Finally, there is need for transparent communication of methodologies and findings and protocols. So always remember to publish open methodologies, findings of the use cases for all stakeholders of the omics ecosystem. This can promote trust and collaboration among stakeholders for inclusive and innovative omics approaches, the uptake of new methods, and the use of data produced by previous studies. So this is a common effort that EFSA, but also all the other stakeholders should remember to help everybody to advance on this. This is the end. I will leave the floor to Santiago for the really conclusion of the roadmap before the Q&A.
Okay. So we believe that this roadmap will guide EFSA in prioritizing projects, and it is really well aligned with what was discussed in the Omics theme concept papers. So we have found out that today the Omics theme concept papers and the work done in that moment is still really relevant. And also this highlight that still needs to be a continuous work to go in through the EFSA vision of the integration of omics. And we believe that EFSA could enhance, thus the application of this proposal, the omic integration in risk assessment. And this will be supporting a linear, automated, and mechanism-based approach to food and feed safety. And we, through the project, through the different recommendations, and through the discussion with the different stakeholders, we have identified several areas of integrations that it was reflected in the proposals. And these include the harmonization approach for third-generation sequencing, the promotion of the usage of underexploited public databases for risk assessment, the adoption of metagenomics in risk assessment, the usage of omics profiles for comparative assessment, the evaluation of proteomics for allergenicity assessment, and the facilitation of omics analysis and interpretation for EFSA and external stakeholders. However, we also found that different levels, but there are several horizontal challenges that need to be fulfilled, like standardization, data access, regulatory issues, many of the ones that was already highlighted. But importantly, we find that these challenges are common across agencies. And this highlights there are obstacles that need to be faced in adopting omics in regulatory science. And that is why we believe that EFSA is in a position to promote and foster the collaborations to work together in the advancement and tackle of this challenge of the implementation of omics in risk assessment and regulatory science.
Thank you very much.